conducted via smooth muscle contractions. So that's kind of why we say smooth muscles are like involuntary muscles for the most part. And so let's kind of look at the types of contractions. Like I said, there's not one single type of contraction I can summarize for you for smooth muscles. There are multiple types here. And it kind of matches the job it needs to do, really. So one type here is phasic. The idea of phasic is without excitation, it stays at some basal level, but as you excite the smooth muscle, it kind of peaks its contraction. Basically, like a skeletal muscle, it does a twitch and then returns to some constant level of tension, right? And you can kind of see something like this, where as long as you give it a pulse every once in a while, you can have this kind of oscillating tension here. And that's why we call it phasic, because it can have phases of tension and not tension, as long as you kind of give it an intermittent kind of excitation, basically. And you would see this something like in the GI tract, right? We have peristalsis. Hopefully you learned that in anatomy, where you have waves of contraction that push food through your GI tract, right? It's basically these contractions that kind of travel down the tubes of your GI tract. And the way you see that is basically through phasic contractions, right? The smooth muscle along your GI tract will do phasic contractions, so you can have a traveling wave of tension, pushing food down the GI tract. And then we have tonic. So hopefully tonic is a familiar word. Basically, tonic means the level of contraction in your muscle is dependent on how much of a signal you give it. So, for example, you can have a constant level of contraction, and then you can take away that stimulus that like dips the contraction, and then it'll turn back to that constant level. Example of this is basically vascular smooth muscle. So a smooth muscle found in your circulatory system, it's going to tonically vary its tension depending on what you need. So do you need a higher blood pressure? Do you need more of a more flow? It's going to vary its tension tonically depending on what you need, what kind of constriction dilation you need. And so these are two types of contractions. Even the way smooth muscles communicate can be different. Some muscles are called single unit smooth muscle cells. And that's because all the muscle fibers, this muscle fiber and this muscle fiber, each one of these muscle fibers are connected by gap junctions. Come on. So each one of these have gap junctions, and we talked about gap junctions before. Hopefully you remember from anatomy. You've seen it again in local signaling of cells. This is an example one. You have gap junctions that they can share the cytoplasm from one cell to another. This way, they can share basically depolarization of one cell to the other cell, right? So if you depolarize, say, this cell here, the cell gets depolarized. A depolarization will then travel to other cells. It would go this direction or go this direction, and also depolarize other cells. So that way, all the muscle cells in the smooth muscle tissue will get depolarized at the same time. And so, when these varicosities, stimulate just this top layer of muscle cells, these guys also feel it too. Then you have multi-unit muscle cells. And so this is the idea that each muscle fiber has to be stimulated separately by its own varicosity. That each muscle cell kind of works on its own, and they have to rely on a varicosity stimulating itself. And it's not going to talk to other muscle cells like you see in the single unit smooth muscle. And this way, this is another way, there's multiple ways of doing this, but this is one way that smooth muscle tissue can get gradation of tension, right? So this is the idea: if you can tension, you can have gradation here by activating, 1, 2, 3, or more muscle cells. So if you just you just excite one muscle cell, you have a certain amount of tension in the tissue. But if you excite another one, you have even more tension. If you excite another one, you have even more tension. So that kind of gradation of how much tension you want is what we call a graded contraction here. And that's the benefit of having multi-unit muscle cell unit here, is you can have that gradation and contraction here. This is how you can dilate or constrict your pupil at varying lengths here, at varying diameters, is by stimulating a certain number of muscle cells. You'll find out later that even one muscle cell by itself has graded contractions too, and we'll see that later. Like I mentioned before, there are no sarcomeres. So this is what a sarcomere looks like in skeletal muscle. This is also what it looks like in cardiac muscle. Cardiac muscle. But you don't have that in muscles, a smooth muscle. This is kind of what it looks like. It looks similar, but what these thick and thin filaments are attached to are these dense bodies. So these dense bodies are part of the cytoskeleton. So the cytoskeleton, remember, is basically the skeleton made of intermediate filaments and other types of filaments that give structure to the cell. And so along the cytoskeleton, there are these dense bodies that attach the thin filaments, and then between these thin filaments, you see these thick filaments. And so what happens is when cross-bridge cycling happens, these dense bodies basically get pulled together, because what's linking them together are these thin filaments, and these thin filaments are basically sliding together as cross-bridge cycling is occurring. So you don't really have unidirectional contractions like you did in skeletal muscle. Skeletal muscle basically went from here, it did this motion, right? Well, now this motion is happening at multiple axes because they're no longer lined up from left to right, but now all the thick and thin filaments are splayed out kind of radially from all the dense bodies. And so when they contract, it's kind of like basically a sphere getting smaller. Now they're not exactly spherical, but that's kind of how you can think about it. In all directions, they're getting kind of smaller. Rather than just a tube kind of shrinking along its length, now it's just kind of like a sphere shrinking its radius. And so that's kind of how smooth muscles contract as a whole cell. They also have connective tissue that connect cell bodies, dense bodies from one another. So as dense bodies from one cell contract, they can kind of pull on other dense bodies. All right, so how do you actually get smooth muscle cells to contract? And so the trick is, it's all about calcium here. Calcium is released into the cytosol not just from the sarcoplasmic reticulum here. So here's the sarcoplasmic reticulum, and just like a skeletal muscle, it releases calcium into the cytosol. And so, you'll see calcium is a key factor in getting a contraction to start. But you also have extracellular calcium coming into the cell. So we have two sources of calcium. Calcium coming from the ECF, and we have calcium coming from the SR. In fact, sometimes extracellular calcium can induce the sarcoplasmic reticulum calcium release. So often one will lead to the other. Not always. This is not always a guaranteed thing. There are some smooth muscles that don't always operate like that, but in some smooth muscles you can have that. And so it's all about kind of controlling how calcium is kind of let into the cell. And how that goes about is really varied. You can have ion gates that get a chemical signal that open up, that lets calcium in. You can have a depolarization event, which you can have voltage-gated calcium ion channels that open up. You can even have mechanically gated calcium ion channels that open up if the muscle cell feels a certain amount of stretch. You see that often in smooth muscle cells around blood vessels. And so there are many ways that you can have these calcium ion channels open up. You can have chemical, mechanical, voltage, and it's going to vary depending on where you find the smooth muscle. But one way or another, calcium gets into the cytosol. One way, somehow, it gets into the cytosol, and by that also induces the sarcoplasmic reticulum to release its own calcium. So you just have a whole bunch of intracellular calcium floating around. Now from there, calcium attaches to this protein called calmodulin. So calcium binds with calmodulin to make this kind of complex, this calcium calmodulin complex. And that's an important complex because this will this will basically act as a second messenger to do this amplification event. So this kind of looks kind of like cell signaling, in the sense that this calcium calmodulin complex will set this basically this signal pathway. It will then take an active, what we call myosin light chain kinase. I'll write that out for you. Myosin light chain kinase. It will make it inactive to make it active. And so, what does a kinase do? We talked about kinases before. Not removes, it adds a phosphate group to stuff. And so this kinase is going to go and basically add a phosphate group to myosin heads. That's this part here. So it's going to go find inactive myosin heads, attach a phosphate group to them, and that makes the myosin head active. And then when that